There will be two introductory talks about neuroscience, one today and the second tomorrow. And because of the great challenge that we have to understand our brain and mind, I decided to split it in two portions. One is very simple, I would say primitive, introduction into main terms and concepts of modern neuroscience for those of you who are not familiar with neurobiology. This will be today, and this is the brain as we know it. The second talk tomorrow will be addressed to big unsolved questions in neuroscience, which wait for their discoveries as we believe in this school with helps of physics and mathematics. But I will start today also with a short teaser with the questions: why brain, why physics, and why mathematics? And I would like to start with a story about a great philosopher who was also a great mathematician and physicist, because at those days these issues for scientists who were thinking deep or not separated. That was René Descartes. I will tell you a story about Descartes' secret notebook. But first about Descartes and physics and brain. There is a story that once Descartes' friend asked him what kind of books in physics he prefers to read. Descartes took him by hand and led to a small house close to his living room and living house, and opened the door, and there were tables with the bodies of dissected animals. He was doing these dissections, buying the animal bodies from butcheries from different places and looking into what is inside. And he said, these are my favorite books in physics. So this is one of the issues of linking physics and the brain. And he was dissecting these bodies, also with a hope, main hope, to understand how our mind and our soul, for Descartes it was the same, mind and souls were synonyms, are getting into contact with our brain. About secret notebook. When Descartes died, and he died in Sweden, he was teaching the Swedish princess at the time, and he died from pneumonia suddenly. His archives were sent by a ship to his friend and editor who lived in Paris. And from the port, they were transported by a boat through the Seine into Paris, into the home of his friend and editor. But something terrible happened. Due to instability of the boat, it turned and all the boxes with the Descartes' notes and his archive drowned. It was a terrible loss. But then a miracle happened. 3 days later, somehow this huge box with all his papers reappeared on the surface of the river. and his editor collected them and saved them, so they were finally saved. The story goes on. 17 years later, into the door of the house of this nobleman, there was a knock, and he opened the door and saw a young man. This man with a German accent said that he is a German diplomat, and for him it is of vital importance to see the notes of the card. He was waiting and searching for these opportunities for many years. So this is one of the drawings of the card where he tries to show how the information from outside gets into the brain and reaches the place marked by H, where he believed our soul or our mind gets in contact with the brain matter. That young man who was trying to find the Descartes archives was Gottfried Wilhelm Leibniz, another great mathematician and philosopher. He came as an ambassador from Germany to serve in Paris, but his idea was to find what Descartes discovered about the contents of the brain and mind. And he pleaded a friend of Descartes to show him these archives. He looked through these archives and discovered that Descartes, among other things, unpublished things, had a secret notebook where he was putting the notes on most secret thoughts that he had. Many of the pages of this notebook were encoded. And Leibniz started as fast as possible copying this notebook, because he had both to copy it and to decode it at the same time. And he managed to copy only 1.5 pages of this secret Descartes notebook. And then the keeper of the archives, who was very jealous about what he was doing, stopped this and didn't allow to continue. Few years later, this secret notebook of Descartes disappeared forever. Probably it is still alive in some areas of France among other religious writings, but we do not know. For 4 centuries we haven't seen it. And 1.5 pages of the secret notebook, which were copied by Leibniz, is the only thing which remained from his secret thoughts of Descartes. And on one of these pages was this drawing of the brain, to which Descartes paid obviously very high attention and importance. You can see the brain seen from the bottom, and you can also see the point there, which is hypothalamus. For some reason, it was marked with special attention by Descartes. Leibniz himself had his own theory of how the mind is represented and gets in contact with matter. He introduced the concepts of monads in systems with memory. He wrote, monads are elementary units of soul. So that was his decision. We will come back to Leibniz's idea and the famous Leibniz-Mill argument about insufficiency of understanding brain to understand mind in the second lecture at the challenges which are still remaining. But there were many other physicists who continued through centuries to try to understand the relations between the matter of the brain and our mind and consciousness. One of them was famous physicist of the 20th century, Schrödinger. Most of us know his famous lectures, which were published later on as a book in 40s, What is Life, which inspired a number of physicists again to go into understanding of units of genetic inheritance, of genes. But later on, 10 years later, he wrote another book which is called Mind and Matter, where he tries or attempts to bring contacts between physics and science of mind, or neuroscience. And he introduced the hypothesis that material events which are related to consciousness are through mechanisms of learning in the brain. This is very important hypothesis. Now, 400 years passed from Descartes' dissections. How the brain looks like today. This is the picture of the brain. In this case, it is mouse brain, which is seen from the same point of view as Descartes drew his secret notebook painting. You can see it also from the bottom. Hypothalamus is there, but the difference is that this brain is optically transparent. It is through experiments in which we develop techniques to clear the brain, so it becomes optically transparent, but keeping the staining of individual cells in the brain. And here you can see the internal structure where individual nerve cells forming the brain network are visualized through their fluorescence in a special type of light sheet microscope. So we now can see with cellular resolution the whole brain and its elements, something which Descartes would have loved to see in his dissections. This is another view of the same brain, and you can see that we can not only make dissections, but we can make a volumetric reconstruction of the brain, and you can see here individual neurons and their neurites, about which I will tell a bit later. But mystery remains. We see the internal structure of the brain, and I will tell now about its elements. But we still do not understand where the mind and consciousness is and how it is implemented and appears through the activity of the brain. And physics and mathematics, we believe, the organizers of this school, it might be a wrong belief, but we still believe, will play a critical role in understanding. Why? First, because as Albert Einstein said many times, we understand a number of natural phenomena in case we have a theory which can encompass and explain them. So in order to understand the huge amount of neuroscience data today, and each year there are about 300,000 publications in neuroscience. Their overall amount reached several millions. No one is able to read all of these papers, and not a single experiment will give us understanding of what the brain is doing and how it works. We need a theory. And making a fundamental theories and discovering fundamental laws has been the great advantage and talent of theoretical physics, which worked well in many fields, including in 20th century molecular biology. We hope that these fundamental laws, which still has to be discovered, will come through the interaction and complex of neuroscientists and physicists and mathematicians. And second, the neuroscience then will enter a new mode of operation. What do I mean? So far, neuroscience is an empirical science. We do experiments, make observations, discover some new facts, and are trying to build understanding based on these facts. If neuroscience will enter the phase of theories, then the mode of operation becomes, as in all theoretical based disciplines, different. The theories make experimental predictions, which have to be tested in experiments. This is other type of experiments, testing the predictions from theories. And theories are often, and in neuroscience we know it well, are far ahead of experimental techniques. This is the same actually in physics. If you will think of predictions of the general theory of relativity and how many years has passed to test some of them, very similar thing will be in neuroscience. When we will have theories, they will predict things which are still not possible to measure experimentally, because we didn't yet develop the tools to look for these predictions. And for this, we will need a phase where experimental physics will be very important, because experimental physics in the recent decades made a tremendous progress of contribution to techniques in neuroscience, starting from magnetic resonance imaging, positron emission tomography, to recent techniques in optics, laser physics, which allow us to look into the brain, like I showed you, for example, for the dead brain, and also into the living brain. But these are new techniques which require experimental physics. And this process is, at the moment, very active in the body. Here are examples. For example, Simon's Foundation, which is advancing research in basic science and mathematics, according to its slogan, holds the special program, which is called Global Brain, where many outstanding mathematicians, physicists, and neuroscientists are studying how the brain works, funded by Science Foundation. There is a whole new discipline called neurophysics. And there are several groups and labs in the world which work in this field. And you can see how it is defined. It can be viewed, what is important, as an approach to neuroscience that is based on solid understanding of fundamental laws of nature. So we are looking for fundamental laws of nature of the brain and mind. There are different activities, schools, conferences in neuromathematics, in geometry of neuroscience. So this is our field today. But before going to basics of neuroscience terms, I would like to mention a different part of our problem, which is still very rarely discussed, even in the groups of people who work together in the theoretical physics and neuroscience. It is the way how our progress, fundamental progress in understanding the brain and mind and consciousness can contribute to our understanding of number of big issues and problems which are in fundamental physics. Here I listed two such questionnaires raised by two theoretical physicists, our famous physicist Ginsburg, who named three great problems in physics, and another legendary theoretical physicist, John Archibald Wheeler, who also made the list of what he called early Q, really big questions. And you see that these questions are: how come existence, why the quantum, participatory universe, his special term to link what happens in the world outside with the observations of this world. What makes meaning, and his famous problem or question, it from it, existence from information. Another theoretical physicist, von Baer, added two additional questions a few years later. What is information, and it from it? And just as a teaser, with these arrows, I listed tentative questions which can be linked to neuroscience and require our understanding of how our brain and mind work. So because of all these challenges, as Alexander Sergeyevich already told 2 years ago, we started a kind of initiative in Russia, which we called FISMART Brain Initiative, which attempts to bring together strong traditions in physics and mathematics in Russia with neuroscience and problems of brain and functions. It is, of course, internationally faced initiative, so we invited also a number of Internationally known scientists who already study and has done a lot of advances in physics of the brain in recent years. And we hold this first conference a couple of years ago, and our study is just a continuation of this, and we are very glad that many distinguished scientists who are pioneers in the field of neuroscience and physics and neuroscience agreed to give the lectures. So now a few key terms and ideas of brain as we know it today. First, no one will be able to give an overview of the brain in one lecture, but there are excellent textbooks in neuroscience which you can address and consult and study. Here are four such textbooks which are most commonly used and with which neuroscience is studied internationally. This is Principles of Neuroscience by a group of distinguished scientists led by Nobel Prize winner Endel. Each of them has passed through many editions. Fundamental Neuroscience, coordinated also by a group of neuroscientists and editors led by Larry Squire. Neuroscience, 6th edition now, by Dale Burns and colleagues. And the 5th edition I put here because you can go to the website of the 5th edition as the publishing house. And there is not only the book, but different animations illustrating different basic principles and ideas of neuroscience. So about brain. The main functional unit of the nervous system is a single neuron. Here you can see staining, histological staining, made by different stains of a part of the nervous system containing nerve cells. These two stains stain the bodies, and this stain, famous border stain, stains not only the bodies but the whole cell with all its protrusions, neurites. They are called neurites. This is the staining of a part of the cerebral cortex. So our cerebral cortex lies on top of the rest of the brain. It is a very thin layer, so you can, if you will expand it, it will be like a tablecloth, but it has very uniform structure organized vertically into different layers. And here you can see the cells belonging to different layers. What neuroanatomists did in the 19th and 20th century for a long time is to study what was called cytoarchitectonics of the brain by looking at the difference of how different parts of the cortex have the number of neurons, the size, type of neurons in the different areas of the cortex, which is the seat of our human knowledge, mind, understanding, and higher brain functions. When the Borges stain appeared, discovered at the end of the 19th century by Italian anatomist Camillo Golgi, there was a revolution in neuroscience. through the studies of Golgi cells and many other neuroanatomists who appeared with this technique, and most notably famous Spanish neuroscientist and neuroanatomist Ramon Cajal. Both Golgi and Ramon Cajal received the Nobel Prize for these discoveries of the structure or organization of different nervous systems. What Golgi staining does, it stains selectively some of the neurons, making it impregnated with a special silver technique, and making them dark compared to others which are not red. If you think that this is a column of the cerebral cortex that contains all neurons stained with this technique, you're wrong. For some reason, the staining selects and stains only approximately 1 of 100 neurons. And it is still not clear enough for what reason this one neuron is stained and other 99 neurons around are not stained. There is special features of the neurons which are stained, but still it stains unselectively different types of neurons. So what you have here is a representative picture of a part of the cerebral cortex. But if you would like to see how all this part of the cortex looks like, you should multiply this by 100.
Excuse me, could I ask a question?
Yes, please.
I'm not sure I completely understood the difference in these three pictures. I mean, as I have understood, you said that this is pictures of our brain core, mainly of neurons. And I want to ask a question. Could you repeat, please, what is the difference between these pictures? Like, you said that, as I understood, that there was a difference in the way of thinking or something else?
No, these are three different histological states. Histological states, chemical staining. So these are dyes, if you would like, which can stain cells. You put the section of the brain in a certain set of solutions, and some of the neurons became consistent, or some of the cells. And here you can see the way it is stained, where the dye is staining mainly cell bodies, and here it stains in the singular part of the cerebral cortex, individual neurons with their protrusions, which are called neurites. Is it clear?
Yes, but I want to understand, actually I would like to understand in more detail the processes between neurons. I mean, I see on this picture that the last one, like I can see that there exist more connections between neurons, and I would like to understand what that
Let us draw it. Unfortunately, I cannot use my notebook because I would have otherwise showed you more pictures of what is going on. But schematically, these are three different neurons, nerve cells. They are lying in the brain section, which you get by cutting the brain on a special device which is called microtome. So you get very thin, several micron section where several layers of neurons are present. But if you will put it under the microscope without staining, you will see very few things because you will not see the contrasted cells. They will be not stained. So people apply different type of dyes. They put this section on the part with a piece of glass into the solution, which stains the section. And one of the stains will stain you only cell bodies. And this will be pictures 1 and 2 with slight variations. Another stain, which is the third border stain, will show you all the minor details of a single neuron with its neurites. And the difference will be that it will show you not all the cells. It will show you this cell and probably this cell. But for some reason, it will not show you this cell, other cells. But you will see the snapshot of the structure of neuronal architecture of the brain with a gorgeous stain. But things get more complex. This is the modern state. It uses novel technique, which is called brainbow. And the technique uses genetic engineering and introduction in the genome of animal specific genetically encoded fluorescent proteins. What happens with this is that now the cells of this organ, this is mouse, in their DNA will have foreign genes which are not present usually in the body of mammals. These genes encode fluorescent proteins. They were received and cloned from different, for example, deep sea organisms which show fluorescence as a part of their life and activity. And by molecular biologists and genetic engineers, they were cloned, and it is possible by genetic manipulation to put them into DNA of the mouse or other organisms. What will happen then is that when these proteins will be expressed among other proteins in the cell, if you put the cell under a specific illumination, fluorescent illumination, they will shine. Therefore, they are called fluorescent proteins. So you will be able to see the cell which expresses these proteins under the fluorescent microscope as a colored cell, like here, for example. This is if you have only one fluorescent protein, for example, green fluorescent protein, all the cells will be green. But what rainbow clever techniques does, it introduced into the genome several, I need to use here. Several fluorescent protein genes. One is red, second is green, and the third is blue. And then interesting thing happens. The cell starts to synthesize these proteins in different proportions. So some cell synthesize more green protein, and some synthesize more red protein. And in combination, when you light the section, each of the cells has different color as an RGB palette in TV. Because of the mixture of three different colors, you receive a unique color. And this is what you see here. These cells are different in their color because of the different combination of fluorescent proteins genetically engineered into them. What it makes possible is, first, here you can see comparable to Golgi staining, for example. Here you can see all cells, all neurons. This is part of the mouse brain, which is called dentate gyrus. It is a part of another structure, which is called hippocampus. I will show you later on an animation video about it in the mouse brain. But what is important to know is two things. First, this structure has an intimate relation to our memory. And the way the memories are being encoded and stored in the mammalian brain, including our own brain. And second, this is a part of the brain which has probably the densest, or one of the densest, along with the cerebellum, package of neurons in a volume space. These are small neurons which are called granular cells, and they lie very tight to each other. So the mouse dentate gyrus, very small area, contains about 400,000 neurons. What this new technique allows is exactly what you asked initially, what you are interested in. It allows you to first level with unique colors individual cells. And second, again, I do not have presentation on this notebook, but it is possible to trace the neurons of each of these cells because they are also colored. It is much more difficult. Here you can see all the borders, but you might start asking how they connect to each other. And for this, you know from which neuron what neuron is. And by coding them by different color, you can start to understand how they organize into cortex, into bundles, how they make connections. Yes.
Am I correct? Each neuron has a special color or has genes of colors.
Each neuron is displaying different color under fluorescent illumination because it has a combination of different number of molecules of free protons, RGB. So if it expresses for some reason spontaneously more red protein, it will be more in the red domain. If it is more red and blue, it will be violet, etc.
Is it known why singular neurons produces different proteins in different portions, even like cells are therefore to each other?
There are different reasons of the regulation of transcription of the genes because they are subjected to modifications of chromatin, epigenetic regulations of how these portions of DNA are opened or closed during the individual development of neuron, which, if you would like, traces the history, the fate of the differentiation of this cell. So therefore, in one neuron, one gene is more open to expression, to transcription and translation of more protein. And in the other neuron, this part of the chromatin is more closed, and the other is more open. And you can see the number of neurons which are contained in different nerve systems. Neurons to the brain are very similar to what genes are to our genome. They are individual units of the nervous system. But with the genomes, there were many surprises while sequencing the genome. It appeared, for example, that humans, big humans, have much fewer genes than were expected. Initially, molecular biologists in the 70s or 80s believed that we have about 100,000 or probably 200,000 genes. And after sequencing, it appeared that we have to be much more modest. And first, it was thought that we have 32,000 genes, and now we're down into 20,000 genes, not very far from the mouse. And certain plants, for example, have much more genes than we do. With the brain, it is more straightforward. Simple nervous systems, there is a term simple nervous system of invertebrates, have few neurons. One of the famous model of organisms in neurobiology is a small worm, nematode, which is called Synaptic Spines. In a heroic study, which took many years by a group of Cambridge scientists, they cut the nematode with a very thin microtome, which allows to make them electron microphotographs and made it serially. So they collected each section of this nematode. And then, and it was in the 80s when there were no artificial intelligence techniques to make a reconstruction. They mainly reconstructed from one section to another the contour of each neuron, so that finally they came with the first map, which was called later on monochrome. You will see and hear today the lecture on Professor Paul Spons, who was one of the scientists who introduced this idea of monochrome. Monochrome is the full graph of connectivity or matrix of connectivity of cells in the nervous system. So the full monochrome of the first organism which appeared was 15 or more years before the idea or the term monochrome appeared. It was the monochrome of in neurons, but their synapses connections are traced in this study. So there are about from 6,000 to 8,000 connections between these neurons, depending on whether you count, I will show later, only chemical synapses or electrical synapses as well. In another, yes.
Can I ask maybe a philosophical question, but I suppose it's very fundamental. Can we have structures in the brain that can that cannot be understandable by our senses now? Может быть, просто чувство, ну, просто заметить то, что может быть.
Brains are part of biology. The universal principles of biology is that we have organisms which are made of cells. They are either unicellular organisms or multicellular organisms. In case of the organisms with brains, these are multicellular organisms, and their brains is a collection of different cells. These cells are neurons, but not only neurons. There are different other types of the cells. For example, glial cells, which is a growing topic in neuroscience because it was thought that glial cells lying between neurons are only metabolic and supportive structure. But now it becomes more and more obvious that glial cells take place part also in information processing, especially small information processing related to plasticity and memory. But nevertheless, you can view the brain as a collection of many cells, and you can count the brain as the organ and system connected cells. There are also blood vessels, of course. And with the resolution of this scale, not molecular, but cellular, I would say that we can see everything in the brains of starting from very simple organisms like a worm to very complex organisms like a human being. In this respect, there are no mysteries. There are no unseen structures. If we will go lower at the molecular level, there are possible molecules or molecular structures which are not yet discovered, but we are again with the increasing resolution of microscopy and super resolution microscopy are getting, and the electron microscopy are getting more and more of this. So my guess to your question would be that there are no surprises on the structure that composes the brain at the cellular level, or even the molecular complexes like synapses, the contact between the different cells, etc.
Если бы сейчас человечество не было условно зрения, да, восприятие жизни, как мы это понимаем? А что сейчас мы просто еще человека, который способен восприятия?
It's a question to the challenge about observations which I mentioned in physics and the real world which is outside. We can sense what is happening in the world. only through our senses, physiological senses, vision, for example, olfaction, hearing, etc. Whether there is anything else which is not powered by the energies which we use in our perception. First, we have the extension of this perception through different instruments, which translate different modalities of the spectrum into our vision. So we can make visible the things which are not sensible to the eye, naked human eye. Second, I don't know what you mean by invisible things, and this is the difficulty. So perhaps there is something which we are not able to sense. Yes, probably it is 95% of the brain, which is like dark matter and dark energy not seen. But there are no predictions from the current neuroscience that such dark matter exists in the brain, which is not, which shows itself but not visible through our instruments today. Mouse brain, with which many people work, contains about 70 million neurons, and our human brain contains about 81 billion of neurons. It's a huge number. So here you see again the pyramidal cell stained fluorescently, and this is the structure of the cell, which is important for us to know. An interesting story about Cajal and Golgi is the fact that they used, it's also about measurement, they used the same technique, Golgi technique, to study the organization of nervous system. But Golgi believed that the brain is organized as a syncytium. That means that it is a net without discontinuities. It is a continuous net. And Cajal, looking in the microscope with the same technique, has seen this network as contained of individual cells. That was a basic idea of the middle of the 19th century, that the cell is an individual unit of organism, and he saw this in the brain. And he provided a number of arguments for individuality of cells in their development, in their pathology, etc., etc. When they received Nobel Prize in 1906, they started, especially Golgi, during the Nobel Prize lectures, to do what was not allowed and is not allowed in Nobel Prize lectures. They started to make a scientific discussion, to argue against each other positions. But Cajal appeared to be fundamentally right. We have the neuronal network containing individual cells, and the cell consists of the cell body. It contains nucleus with the genetic apparatus DNA, like other cells, which produces involves different proteins which make the phenotype of this cell, how it looks. And usually, the typical cell looks like this. It has a central body. It has one long protrusion, which is called axon, by which the cell sends signals to other cells in the network. It has a particular part of the cell body which is called axon hill, where this electrical signal is usually generated to spread in this direction. At the end of the cell body, of the axon, there are so-called axon terminals. This is the places where this cell contacts other cells. We will speak about this at the synapse. And on the opposite part of the cell body are neurons which are called dendrites. These are protrusions at which the cell receives signals from other neurons. So you can use the neuron, the simple neuron, primitive neuron, as getting the signals from many other neurons and giving, synthesizing them and giving its own decision and response in a form of electrical signal to other neurons. This is the typical picture of the cell body, and you can see that it contains mitochondria, ectoplasmic reticulum, etc. Very similar things to other cells in the body. I should say that I emphasized twice or thrice that this is a simplified picture. Why? Because sometimes the signals are generated not in the axon, but already in dendrite. And because sometimes, and probably quite often, the signals are sent not only through axon, but through dendrites also. So dendrites can send signals to the neurons which connected to this neuron and gave signals. There are reciprocal connections and signaling. They are called the reciprocal synapses. I will draw it a bit later. The great advance in neuroscience and neurobiology was made in the middle of the 20th century through discovery of the way the neurons are generating electrical signals. The main idea is that the membrane of a cell, of neuron, contains proteins which are called ion channels, and they have selected permeability to different ions, and concentration of ions inside and outside of the cell is different because of this selective permeability of ion channels. These ion channels, which are shown here, are regulated from outside and from inside of the neuron through different type of receptors. So this allows to change the permeability of ions and to generate potentials, changes in the potential difference between concentration of charged ions in the cell. And this gives finally the cellular action potential, which I mentioned previously, when the cell is able to fire a signal sending an information to the cells which are in contact with its axon. Action potential has a particular shape and dynamics. I will not go into this, but this allows, which is important, to record the activity of the cells both electrically by using electrodes, which record difference in the potential, or optically, the special dyes which can be potential sensitive dyes, or calcium recording, which you will use in intracellular signaling accompanying the action potentials, which you will use in some of the of your projects. Yes, please.
Maybe it's a preliminary question, but I think it's connected. You said that each neuron is included into a network, but I just don't know how, for example, our hormones influence on this network. Like we have different hormones, for example, adrenaline. I don't know how it's called.
Yes, it will come later. Yes, they do influence, and a lot of what is going on in the nervous system is chemistry.
And do they influence on
Yes.
their electricity?
Yes, yes. Through specific type of receptors, which sense the chemical molecule. It can be a chemical molecule secreted by one neuron and sent to another, but it can be a hormone also coming from the blood, and it can be an intermediate type of chemical molecules, which are usually called neuromodulators, which are released by neurons, but are spread diffusely, not addressed to particular neurons, but in all the surrounding of this terminal. And they influence different neurons, but they influence the membrane potential of these neurons or the specific acting on specific receptors on their membrane. So here is the experiment showing you how going from the action potential, you can look into the activity of the brain. What is shown here is the experiment where the mouse is implanted with a set of microelectrodes. Each electrode is a thin wire, usually about 30-50 microns. Sometimes these are glass microelectrodes, which are much thinner, about 1 micron, but it is more difficult to apply them to the human membrane, to implant this glass microelectrode. So usually the platinum iridium thin wires are used. By implanting them chronically into the brain of the animal, you can record the electrical activity in the vicinity of the tip of this wire. It can stay for a long time, in monkeys, for example, for many years, and you can listen to the recording from these electrodes, how the neuron in the awake animal brain behaves, and in the network of other neurons during certain relations of this organism with the brain, with the environment and the objects here. And there are very interesting data emerging from these recordings, which were pioneered and developed first in 1950s, but now are very advanced with the new nanotechnology techniques. Here you can see the mouse, which explores a box in which there are two dishes. And for the mouse, these dishes resemble a nest. So the mouse can hide inside the dish, and it perceives it as a nest. And there is a concept of the nest in the mouse. And though this is not typical nest which you will find in nature in the mouse inhabitant, the neurons in the mouse brain are sensitive and responsive to the moment when the mouse is hiding in this dish, perceiving it as a nest. So each vertical line here is one action potential of a single neuron recorded with the microelectrode inserted in the brain. And this is dynamics of spiking action potentials of this neuron while animal is doing its behavior. So you will see here that the mouse is approaching this dish, which is a concept of nest, or a symbol of nest. It looks like nest. And this neuron will start firing. It was silent before, when the animal will get into the nest. So this neuron recognizes nest. Out of the nest. This small box is not a nest because you cannot hide inside. This is another nest. So we can use the electric potentials of individual neurons to start to understand how the whole brain consisting of this neuron behaves in a reasonable manner. Now about chemical transmission signals, molecules, and how the neurons communicate. The central concept here is synapse. Synapse is a name for close contact between two neurons. One of these neurons is usually the one which sends the signal, and another neuron is the one which receives the signal. It was the idea of synapse was introduced by famous British neurophysiologist Charles Sherritt from the end of the 19th century. And it was coming together with the idea of network containing individual cells, which were propagated and suggested and popularized by Ramon Cajal. And so we have cells and we have synapses between them, and we have a network. It was initially believed that the contacts between different cells are electric. So there is an electric transmission of the signal, then it somehow jumps to another cell and continues the electric process here. But beginning in the 20s of the last century, various data started to accumulate, suggesting that something else is happening, and that between the two neurons in the so-called synaptic shelf, there is a chemical molecule which is released by one neuron and is given to another, and there it induces the electrical process. There was a number of experiments supporting this, and finally they resulted in the Nobel Prize and acceptance of the idea that synapses are organs of chemical communication. And in fact, for neurons, it is important not to send electrical signal, but to provide its chemical molecule to another cell, which is a receptor. there was only one famous electrophysiologist, neurophysiologist, John Eccles, who resisted this idea in the 20th century, because he felt that he spent so many years proving that it is electrical transmission, that it will be his personal failure to admit that the transmission is indeed chemical. But he made friends with the famous philosopher of science, Karl Popper, who persuaded him that for the scientist, it is not a shame to get rid of his or her own wrong hypothesis. Indeed, you should kill your own hypothesis by replacing them with the better ones to proceed. So, persuaded by Popper, Eccles was one of the last to admit that there is a chemical transmission between the synapses and started between the neurons and synapses and started to investigate this process so effectively that 15 years later he received the Nobel Prize for the discovery of the mechanisms of chemical transmission in the synapses. And just after that, or soon after that, it was discovered that, in fact, there are electrical synapses in the brain. So the neurons are communicated not only chemically through molecules which contact receptors, but through so-called gap junctions. These are also proteins, special molecules, which form a channel inserted between membranes of two cells. And when there is an action potential here, it can change the flow of ions directly to change the potential of the receptive cell. So when I mentioned that in C. elegans there are from 6 to 8,000 synapses, it is because majority of them are chemical, but some of them are electrical. When we receive a signal in postsynaptic cell, it is called postsynaptic, and this cell is called presynaptic. It can result in several types of processes. Here is the more detailed depiction of this element synapse. So the signal, electrical signal, comes through the axon, and at the end of the axon terminal, there are certain cellular structures which are called synaptic vesicles, which contain, it is like a globe inside, it contains the chemical molecule. The polarization of the membrane at the terminal through calcium channel, specific type of proteins allowing to go calcium into the cell, can bring these vesicles close to the membrane and open them, so that the potential will be translated into the opening of these synaptic vesicles, release of the chemical molecule, and action on the postsynaptic receptors. The molecules which are ion channels mostly, but they can bind a specific neurotransmitter, this molecule, to give a signal either in the changes of the membrane potential or inside the cell. And there are different types of these chemical transmitters. There are, initially, it was believed that there are two transmitters. One is excitatory and another is inhibitory. It is a simple and good idea for mechanical engineering. So you can release one molecule and excite, the postsynaptic cell, and you can release another molecule and inhibit. But in fact, nature in the evolution of the nervous system was organized in a very different way. It used many types of chemical molecules for communication between the cells. It was very ancient. So these chemicals, which are released by one neurons and act on other neurons, appeared many hundred million years ago. And recent studies show that in different taxons, trees of the evolution of the nervous system, slightly different molecules evolved, which sustain communication. Here you can see some of the most common of them. These are molecules which are often described as neuromodulators, as I told you. Adrenaline, noradrenaline, dopamine, serotonin. There is also initial ideas about excitatory transmission or about acetylcholine excitatory transmitter and glutamate excitatory transmitter and gamma aminobutyric acid, GABA, as inhibitory transmitter. But to describe you the complexity of this, just two examples. GABA is classical inhibitory transmitter in the nervous system. It is released by inhibitory neurons. When it is released, it inhibits the postsynaptic cell. But some years ago, it was discovered that during early development of the brain, postnatally, a few days after the birth, in the neurons of hippocampus and probably in other brain neurons, GABA can act as excitatory transmitter. So at this period of the development of neural network, it excites the neuron. Then it switches to its inhibitory action. Can you imagine why? No? This is simple. Everything which determines how one or other transmitter acts is determined not by transmitter itself, but by its receptor. So for one neurotransmitter, evolution has developed many genes encoding different proteins, which are different types of receptors. And depending on what gene for this neurotransmitter, what receptor gene is expressed by this cell, the action of the same neurotransmitter, GABA or noradrenaline or serotonin, will be different. So in order to make the GABA inhibitory instead of excitatory, you have just to change the gene expression of receptors on the postsynaptic cell, and it will become sensitive in a different way. Again, another complexification is that some of these neurotransmitters or neuromodulators, like noradrenaline, dopamine, cannot be described in traditional means of excitation or inhibition. So what I mean is that when you apply, let's say, dopamine to the cell in the living brain, there are certain techniques of microiontophoresis which allow for the glass electrode to push several groups of this molecule onto the neuron and to record the activity. And it appears that there is no response to this transmitter, to dopamine or noradrenaline. But if you will probe the neuron with the application of glutamate, excitatory transmitter, you will discover that there was an effect which didn't change electric potential, but it changed the sensitivity of the neuron to another classical excitator or inhibitor. So therefore, many of these molecules are not transmitter in the simple mechanical sense that there is an excitation here which turns into excitation here, but neuromodulators. They act on the cell in a deep, metabolic, long-lasting manner, changing the reaction of this neuron to thousands of other simple sequence excitator or that are received by this cell. So here you can see different types of receptors. We were discussing that receptors are proteins which are inserted into the membrane of the cell, and two main types of receptors are ionotropic receptors and metallotropic receptors. Ionotropic receptors is when the molecule transmitter or modulator is finding a binding site on this protein, and when it binds, it opens the ion channel of the protein so that ions are passing inside the cell or outside of the cell, changing the membrane temperature. Metallotropic receptors are more complex. First, they have a different structure. They are not forming a ion channel, but they form several loops in the membrane. And in these loops, there is a site which is extracellular, which can bind the chemical molecule. And there is an intracellular site, which can, after binding this molecule, change the biochemical signaling in the cell, molecular reactions in the cell by second messenger. And what happens is that after binding to the metallotrophic receptor, the chemical molecule will change the sensitivity of ionotropic receptors. And therefore, these neuromodulators, without changing the potential of the cell directly, can modulate the responsive sensitivity of the cell to classical classical neurotransmitters. What we have to consider each time when we look at this neural network is that there is no transmission of signals in the nervous system. Why? Because a single neuron, in order to generate action potential, has to integrate the signals from many inputs. So there is no linear linear transmission. If you have several thousand inputs to this neuron, and in order to generate a single action potential, you have at least 100 of them to be activated. And this 100 can come from very different parts of the brain. So a single spike of the neuron is not a transmission from any of these components. It is an integration through the whole nervous system. And there is also an enormous divergence of the signals. How it works, that one neuron will have terminals in various brain structures. So when the action potential is generated by these neurons, it is sent also nearly in the awake, especially brain, by all brain structures. The new techniques of tracing of individual neurons with special viral stains and vectors show that a single neuron can penetrate the full brain, go to many brain structures. And we know for some of the types of neurons, for example, for dopaminergic cells, synthesizing dopamine is a neurotransmitter, that they can act on about 5 million cells in different parts of the brain, both in the cortex and subcortex, in one neuron. So there is an enormous convergence on one neuron and divergence of signals. Furthermore, it can appear through the mechanisms of plasticity that this neuron will send the signal to all its terminals, but not all of them will produce the effect because of the synaptic efficiency of a particular terminal. So there is a play between the receiving neurons and sending neurons, which makes not all of these terminals equal. The portions of the neuron which are receiving signals are often called synaptic spines. Synaptic spine is not a universal type of synaptic structure, but it is a structure which is quite often used and seen in the mammalian central nervous system. It is portion of the dendrite of the cell. So this is one postsynaptic neuron, which receives a presynaptic axon and terminal. Vesicles are here, and here is so-called postsynaptic density. The signal is transmitted in this way to change the membrane potential and can, with the integration with many other such events on other spines, can lead or not lead to decision to produce or not to produce the spike output of the neuron and the axonal unit. I said to you that my description of the nervous system is very elementary, and if you will look at the real structures, you will find many surprising examples. One of the surprising examples is that there are structures which I mentioned and promised to tell more about, which are called reciprocal synapses. These are contacts between two neurons This is postsynaptic, and this is presynaptic. But this is arbitrary because what you can see on electron microphotographs is that here you can see vesicles, synaptic vesicles, which is a sign of presynaptic terminal. So they will receive the signal from action potential and release neurotransmitter to act on postsynaptic cell, which contains so-called postsynaptic density, electron dense number of protein structures which are receiving this signal. But in the same membrane, two membranes, you can see that if you will go along this postsynaptic cell, you will find a portion in the contact with the same presynaptic cell, which contains synaptic vesicles. And here is a postsynaptic density. So here the signal is transmitted in this direction, and here it is transmitted in this direction, in the contact between two cells. I told about metabolic receptors, and here are some examples of signaling cascades that are able to mediate the process of chemical signaling to changes in the long-term properties of neurons through biochemical changes in the cytoplasm and changes in gene expression. So what happens here is that the signal through the second messenger system goes to nucleus, which can produce activation of the specific gene synthesis of mRNA, protein, and finally changes in the structure of this neuron, which is called or neuronal plasticity. For example, in cases of long-term potentiation, can lead to growth of another synaptic contact, which makes the contact between these two cells more efficient. And to finish this introduction, a small video with the 3D reconstruction of what we have seen in our neurobiological favorite model to the laboratory mouse. There it is. This is mouse skull. And here we see the brain with 70 million neurons. There are different structures which I didn't speak about, but this is one hippocampus, which I mentioned. Very important for integration of signals from the cortex and memory. And this is the part which I also mentioned, that digest, that contains dense granular cells for 100,000 neurons. And in this reconstruction, you can see them in a similar way as in the rainbow picture, but in one color. Here, this is our individual neurons, and their neurons. Very much like what we see in the transparent brain project. And these neurons are generating spikes. So you can see how the signals are coming to the individual cells, which start to fire with their actions. This is one cell. These are spikes on the dendrites. These are different dendrites, and this is its axon. It is a small granular cell. The spikes on the dendrites are actually living. They change their shape very regularly. They're moving like here. And to these dendrites coming the axons from the cerebral cortex. These are green axons. And when the axon is firing, it releases through synaptic vesicles neurotransmitter, which is sensed in this case by a moving spike and can lead to establishment of a synapse. Around these are glial cells, about which I didn't speak, but they increasingly recognized as a critical process in formation of long-term memories. And here the signal is going through the spine and comes to the dendrite and giving the action of this neuron within the brain, like for example the nest neuron, which tells the mouse that when the neuron is firing, there is a nest around. The mouse knows that there is a nest because of the firing of this neuron. So thank you very much.